Welcome back. Months ago, we set up our red rose crossbreeding experiments, crosses 19 and 20, and they finally produced a ton of offspring. Our goal was to see what traits they inherited, but cross 20 threw us a major genetic curveball. The half-black trait showed up in a way I absolutely did not expect. Therefore, today we're doing a deep dive on all the results from both crosses to unravel this mystery. For those new to the channel, I'm Ivan. I'm documenting my journey to understand guppy genetics, and I'm using my Snow White strain as the baseline for this now 2.5-year project. But the goal isn't to just keep them Snow White, it's to strategically add new traits, which is what these red rose crosses are all about. This process gets incredibly complex. Therefore, if you want to see the master plan, I've started mapping out past crosses on my website. It has detailed charts and data, way more than I show on YouTube, and will eventually have a store where I'll list guppies for sale. I have a whole list of traits I want to gene stack into my Snow Whites over time. But the traits at the very top of that list are a large half-moon tail and a high fin dorsal. Therefore, the red rose strain, which excels in both, became the key to this entire experiment. In part 1, I explained my full decision-making process for using these red roses. And I did a deep dive on the unique ray bifurcations that I believe are key. Ray bifurcations are the branching in the fin rays. If you haven't seen that video, I highly recommend you watch it, as those concepts are critical for everything we're about to discuss. Now, here is the central problem we're trying to solve. Snow Whites are beautiful, and one of the genes responsible for that white color is magenta. But magenta has a major genetic drawback. It also reduces fin size. This is why my Snow Whites almost always have short, small fins. Therefore, this entire experiment is a massive challenge to see if the powerful fin genetics from the red rose can finally overcome magenta's stunting effect. So, before we get to the deep dive, here's the crucial setup. I needed to test for potential sex-linked traits, genes that are passed down differently by males and females. Therefore, I set up 2 reciprocal crosses on April 3rd. Cross 19 used 4 red rose females and 2 Snow White males, and Cross 20 was the exact opposite, 4 Snow White females and 2 red rose males. But we immediately hit a major setback. As I covered in the last video, I lost all but one of the Cross 20 females, likely due to disease. This is critical, because it means that all the offspring from Cross 20 came from just that one single mother. Both crosses produced several batches of fry, and we ended up with a great sample size. 75 offspring from Cross 19 and 59 from Cross 20. But we immediately saw a strange discrepancy. Cross 19 had an almost perfect 50-50 male to female ratio, while Cross 20 skewed heavily male, almost 73%, with only 16 total females. I'm not sure why this happened. However, even with that odd skew, the total numbers are high enough to give us real statistical power. To make our comparison accurate, I simultaneously set up a pure red rose cross, giving us a perfect control group of the same age to compare against. This also served another vital purpose. This whole experiment could fail. Therefore, maintaining a pure backup red rose line was critical insurance in case I needed to start over. Okay, let's compare the offspring. The first and most obvious trait that jumps out in both Cross 19 and 20 is the magenta gene. You can see this incredible magenta iridescence shimmering across their entire bodies. And when you compare them to one of our pure red roses, the difference is night and day. The crossbreeds are just glowing with it. This result was actually 100% expected. Magenta is a dominant autosomal gene from the Snow White line, meaning it's not sex-linked. But the fact that every single offspring expresses it tells us something crucial. it confirms our snow white parents were homozygous, carrying 2 copies of the magenta gene. Now, magenta isn't the only gene that makes a snow white white. Their full color actually comes from the interaction of 2 more genes, Storsbach and European Blau, and our offspring definitely inherited copies of these. But unlike magenta, both of these genes are recessive. Since the red rose parents almost certainly didn't carry these traits, the genes are completely masked, and we don't see them yet. Therefore, the most important takeaway here is that 100% of these new guppies are now silent carriers, holding that hidden genetic potential for us to unlock in future crosses. So that covers the visible traits from the Snow White line. Now let's focus on the red rose contributions. And the most obvious one is, of course, their red color. But the offspring didn't inherit that deep, rich red. Instead, it's much more of an orange. Look at this comparison. Here's a pure red rose male. Note that distinct, deep red contrast. Now here's a male from cross 19. Admittedly, that magenta iridescence we just talked about washes out the contrast a bit. But the color itself is clearly more orange. This isn't a fluke either. The same orange color was consistent across the males in cross 20. This consistency is actually a really important pattern. If you've been with the channel from the beginning, you'll remember my very first crosses between Gandalf and the original females. They also produced uniformly red offspring. I had a strong feeling crosses 19 and 20 would follow this same pattern, and they did. This confirms that the red coloration is incredibly dominant, especially when those recessive snow white genes are hidden. Phenotypically, these results look almost exactly like we saw way back in cross 4. Therefore, this gives us a new sort of rule for this project. It's safe to assume that any future F1 cross between a snow white and a different strain will likely result in uniformly red offspring. Alright, let's look at one final color phenotype before we move on to fin shape, the half-black trait. This is a key feature from the red roses that the snow whites don't have. And up until now, I've attributed all half-black inheritance in my project to the Nigrocaudatus I gene. But based on the wild results from crosses 19, 20, and an old anomaly back in cross 16, I'm now convinced I was wrong. I believe it's been Nigrocaudatus II all along. This might seem like a tiny semantic difference, but the genetic inheritance mechanism is huge. Here's why. When looking at the gene reference tables, Nigrocaudatus I is listed as strictly X-linked. This made sense, and all my crosses up to this point suggested it was true. But Nigrocaudatus II was listed with a confusing X/Y notation. This baffled me. How could a gene be on both the X and Y chromosome? Wouldn't that just look like regular autosomal inheritance? Well, it turns out 2 things could be true at once, and the results from crosses 19 and 20 will be the perfect way to explain this. Let's look at cross 19 first. The pairing here was red rose females crossed with snow white males. So the half-black trait must come from the mothers, and it turns out 100% of the offspring, both males and females, ended up expressing half-black. This is easy to interpret. The red rose females come from a pure, true-breeding half-black line, so their genotype must be homozygous. X, capital Ni, X, capital Ni. The snow white males don't express half-black at all, so their genotype is X, lowercase ni, Y. Therefore, when we run the Punnett square, we expect all female offspring to be X, capital Ni, X, lowercase ni, and all male offspring to be X, capital Ni, Y. Phenotypically, this means 100% of the offspring will express half-black. This prediction matches our results for cross 19 perfectly. No surprises, yet. Cross 20 is where things get unexpectedly interesting. Let's run the same exercise, using the same assumption that we're dealing with the strictly X-linked Nigrocaudatus I. And in this cross, we had one snow white female, X lowercase ni, X lowercase ni, and 2 red rose males, who we assume are X capital Ni, Y. Therefore, when we fill in the Punnett square, the expectation is crystal clear. 100% of the females should express half-black, and 0% of the males should. So let's see what actually happened. The female offspring, all 16 of them, did indeed express half-black. So far, so good. The Punnett square checks out. But when we look at the males, 33% of them also expressed half-black, when our model explicitly predicted none. This is a little hard to tell in the older males because the magenta color tends to wash it out. But here's a video of the males when they were juveniles. It's clear. Some males are half-black and some are not. This result was a complete surprise, and I had to think about this one for a long time to finally make sense of it. My first thought was that I must be wrong about Nigrocaudatus I, and that I was actually dealing with Nigrocaudatus II. I initially assumed this meant the trait was Y-linked and not X-linked. Therefore, I ran the Punnett square again. Our snow white female remains X lowercase ni, X lowercase ni. But this time, we'll assume the red rose male's genotype is X lowercase ni, Y uppercase ni. This new model predicts that 100% of the males inherit the trait, but 0 females do. This was also completely wrong, as our actual Cross 20 results showed 100% of the females had the trait. Okay, let's review the facts. Cross 19 proves the trait must be at least X-linked. So this leaves just one other wild option for the Cross 20 males. What if they have the trait on both the X and Y chromosome, a genotype of X capital Ni, Y capital Ni? I ran the Punnett square for Cross 20 one more time, using this super half-black male. But this also failed to match the results. This pairing would predict that 100% of all offspring, males and females, should express half-black, and we only got 33% half-black males. It's a bit of a head-scratcher, isn't it? So the key to this entire puzzle turned out to be the specific setup for Cross 20. 2 red rose males and only 1 snow white female. Let me walk you through my reasoning. First, let's look at the pure red rose line. We know half-black must be at least X-linked, because 100% of females express it, making their genotype X capital Ni, X capital Ni. And the males must have at least 1 copy, making them X capital Ni, Y. This setup ensures the entire pure line always expresses half-black. But what if a subpopulation of males also has the half-black gene on their Y chromosome? This would create super half-black males with a genotype of X capital Ni, Y capital Ni. The thing is, you'd never be able to tell these males apart within the pure line, because both male types, when bred to X capital Ni, X capital Ni females, would produce 100% half-black offspring. Therefore, the only way to reveal these 2 different male genotypes is to cross them with a female who does not have the trait, like a snow white female, for instance. And I believe that is exactly what happened. My single snow white female was fertilized by both males simultaneously. The super half-black X capital Ni, Y capital Ni male produced 100% half-black females and 100% Y-linked half-black males. But the regular X capital Ni, Y male also produced 100% half-black females, but 0 half-black males. Therefore, when you combine the fry from both fathers, you get precisely the result we saw. 100% of the females are half-black, but the males are a mix of half-black and non-half-black. Super interesting, right? This is the best explanation I could find that perfectly fits our Cross 20 results. And it finally clarifies what that confusing X/Y notation for Nigrocaudatus II on the gene chart actually means. It doesn't mean the gene is either X or Y, but that it can exist on the X chromosome, the Y chromosome, or both simultaneously, all depending on the specific fish. This incredible genetic flexibility is possible because of a well-known mechanism called crossover, where an allele can physically jump from one chromosome to the other over generations. So what does this mean for the project? Well, it means I was wrong in my initial assumption. I've been calling this trait Nigrocaudatus I, but it's clearly the more complex Nigrocaudatus II. And while the simple X-linked inheritance patterns I described in my older videos still hold true for those specific crosses, this new understanding finally explains the anomaly from cross 16. You might remember a single half-black male appeared where the math said there should be 0. I now believe that was a spontaneous crossover event, and I'll be dedicating the next cross 16 update video to that. Moving forward, I'll be using Nigrocaudatus II to describe all half-black traits. This whole event just highlights the critical importance of setting up reciprocal crosses, swapping the males and females when you combine two different strains. If there are any complex sex-linked traits involved, this is the only way to reveal them. This brings us to the second major reason I set up these reciprocal crosses: to start transferring the impressive fin shapes from the red roses over to the snow whites. I'm especially interested in their tail shape, and my ultimate goal is to eventually create a snow white with a true half-moon tail. As I covered in part 1, I believe the rose tail is the best avenue to that goal because of its extensive ray bifurcations. But I immediately hit a roadblock. I couldn't find any specific genetic information for rose tails. The best data I have is for the half-moon trait. Therefore, I had to proceed with a critical assumption, that the rose tail shape is genetically similar enough to the half-moon that it's inherited in the same way. According to the gene reference table, the half-moon trait is X-linked dominant. This setup should look familiar, as it's just like the Punnett squares we did for half-black. So let's map out our predictions for cross 19. The red rose females coming from a pure line would be homozygous dominant, X capital HM, X capital HM. And the snow white males lacking the trait would be X lowercase hm, Y. Therefore, the Punnett square predicts a simple, clean result. All offspring should phenotypically express the half-moon tail. The females would all be heterozygous, and the males would all be X capital HM, Y, getting that single dominant gene directly from their mothers. To be honest, our Punnett square predicted 100% half-moon, but it's really hard to tell if that's what we actually got. The males are definitely rounder than their snow white fathers, but they don't quite match their pure red rose counterparts. This suggests we might have competing genes at play. Here's the problem. The gene tables list half-moon as dominant, but that might only mean it's dominant over the wild-type genotype. It says nothing about its relationship to other tail types like fan, flag, or delta. And it's very possible our snow whites carry those traits, which have been completely masked by the double dose of magenta that shrinks their fins. These new crossbred fry only have a single copy of magenta. Therefore, its fin stunting effect is weaker, and those underlying snow white fin genetics might be resurfacing to compete with the half-moon trait, giving us this mixed result. This is just a guess at the moment. We'd need more crosses to prove it. So in the meantime, let's move from guessing to quantifying and take a closer look at the tail shapes of cross 19 males. Now, in the last video, I established a quantitative baseline for the red rose tail, giving us specific metrics to compare against. We know they have between 22 and 24 rays, and that the branching or bifurcations start very early, around the 4th to 6th ray from the bottom. The most defining trait, however, is the density of that branching. The longest rays bifurcate at least 4 or even 5 times, creating 16 to 32 individual tips at the end of each ray. But trying to count this on the colorful males is almost impossible. Therefore, we might have to rely on the females. Their fins are more transparent, which allows my camera to actually capture this fine structure. Using this quantitative baseline, let's see how the crossbred offspring square up. At first glance, the Cross 19 males look very promising. They have about 24 rays, with bifurcations starting around the 5th or 6th ray, and their longest rays show about 16 tips, indicating 4 total bifurcations. They even have that characteristic wavy edge. This is all highly consistent with the red roses and seems to perfectly confirm our X-linked dominant prediction. Therefore, if this holds true, the females should express the exact same characteristics. But they don't. The Cross 19 females also have about 24 rays, but their longest rays only end with 8 tips. That's only 3 bifurcations from the base. This is a major difference when you compare them side by side with a pure red rose female, and it directly contradicts our X-linked dominant Punnett square. This result, where the males show the trait but the females don't, is actually what you would expect from an X-linked recessive trait, which would make the females simple carriers. But before we make any new conclusions, let's look at Cross 20. So Cross 20 is the critical tiebreaker. We have 2 competing theories, and they give 2 very different predictions. If we stick to the gene reference table's X-linked dominant model, we would expect to see only the females in Cross 20 express the phenotype, and the males wouldn't even carry the trait. But if our new hypothesis from Cross 19 is correct and the trait is actually X-linked recessive, then none of the Cross 20 offspring should show the phenotype at all, and the females would just be silent carriers. Okay, let's take a closer look at Cross 20. And right away, the males look very different from the Cross 19 males. Instead of having shorter tails, a good portion of these males have larger, almost fan-like tail shapes. But remember, these males also carry a copy of the magenta gene. Therefore, this is even more evidence for our earlier theory. A single copy of magenta does not severely stunt fin growth. This fan-like shape is likely one of those hidden snow white fin genetics I mentioned, which are now resurfacing because they aren't being suppressed by a double dose of magenta. Now, trying to get a clear photo to count the caudal rays on these males was incredibly difficult. But let's go ahead and try with the ones I got. I solidly counted about 21 rays, though it's likely 24. And bifurcations begin around the 5th or 6th ray. Most importantly, their longest rays end in at least 16 tips, indicating 4 bifurcations from the base. This is consistent with the red roses, but the overall shape is that wider fan-like tail. Now for the Cross 20 females. They also have about 24 rays with bifurcations starting early. But, and this is the critical piece of evidence, just like Cross 19 females, their rays also end with only 8 tips. When you see it side by side with a pure red rose female, the difference is obvious. Therefore, this isn't an anomaly, it's a clear pattern. The results from both crosses are inconsistent with the X-linked dominant inheritance we expected from the gene table. However, this pattern seems consistent with our new theory. The rose-tail trait might be X-linked recessive instead. So what's the conclusion here? It's very tempting to declare that the rose-tail is an X-linked recessive trait, as that's what the evidence suggests. But we simply can't be sure with only these F1 results. Therefore, the only way to prove this is to set up more experimental crosses to tease this out, which I'll be doing soon. On a more positive note, this experiment did give us a very encouraging result. We've gathered strong evidence that a single copy of magenta doesn't severely interfere with fin size. This is a huge win, as it means we could be more strategic in our future crosses to make sure that halfmoon or rose-tail trait is successfully passed down. Now, before we plan the next generation, let's touch on one last trait, the red rose's high fin dorsal. The gene reference table is no help here, listing its inheritance as completely unknown, so this was a total shot in the dark. And at first glance, it looks like we missed. None of the crossbred offspring, males or females, inherited that massive flowing dorsal. But look closer. Even though they don't have the volume of the high fin, their dorsal fins are still significantly larger than the original snow whites. This could, once again, be those underlying genetics resurfacing thanks to only a single copy of magenta. Therefore, even though we didn't hit the jackpot, this is a clear win. We've potentially inherited a larger dorsal fin into the snow white line, and that's a huge step forward. Alright, so what's the plan? My primary goal is to continue folding these new red rose genetics back into my working snow white line. But I have a major variable to control. I currently only have males that I am 100% confident are pure snow whites. My available females are genetically messy. Some are half-black, and others are carriers for traits I'm not ready to mix in. Therefore, to limit uncertainty, I'll only be using my known pure snow white males. Based on that, here's the new setup. First, my main two crosses will pair these snow white males from cross 16 with females from both cross 19 and cross 20. This is the official backcross to move the traits into the snow white line. But there's a risk this won't be enough to lock in the fin shapes I want. Therefore, I'm setting up a third cross, pairing a male from cross 19 with a female from cross 20. This will really scramble the genes and will hopefully produce some offspring that truly express the tail shape, even if their overall body color is still a mix. To create a vital backup, I'm setting up a fourth cross. This will be a male from cross 19 back to a pure red rose female. This cross won't have any snow white color, but it will create a crucial genetic reservoir of fish that are guaranteed to carry the fin traits I need for the future. Finally, to maintain my pure red rose line, my fifth new cross will be auxiliary cross 3, a simple pairing of red rose siblings. That wraps up our look at cross 19 and 20. We had a lot to go over, and the most interesting result was clearly the discovery that half-black or Nigrocaudatus II can be linked to either the X or Y chromosome. This isn't just a cool piece of trivia. This new information is invaluable. It's going to be the absolute key to helping us finally understand the anomaly we saw back in cross 16, which will be the focus of the next video. We are making progress towards our goal, but as you can see, keeping track of all these genetic threads is definitely not easy. With these new strategic crosses, I'm confident we can make this happen, or at least learn something new along the way. If you're interested in this genetic-heavy Guppy project, subscribe so you don't miss that upcoming deep dive on chromosomal crossing over events. Finally, let's wrap up with some quick updates on our other crosses. Cross 18. We're finally seeing some snow white males emerge. I initially assumed most of this brood would be dull colored, but a lot of them started coloring up far more intensely than I expected. Cross 21. This cross has already dropped 3 total broods, with some fry just now starting to show their first signs of color. Auxiliary cross 3. Our pure red rose backup line is officially set up, but it's still too early for any fry at the moment. If you're interested in seeing the deep dive into how we started crosses 19 and 20, go ahead and check out part 1 right here. Thanks for watching, and I'll see you in the next video.